Hello, everyone. Hi, Russ.
Hello. Thanks for having me back. For what it's worth, Vladimir, I had the link correct this time. I just had to sign into Zoom. So hello. Thank you for having me.
So maybe let's wait another minute.
Yeah, no worries. That's good. Hey, I was playing around with the API a little bit in VGSim. And I think it's great. At least I can follow the example you had, and it did things that made sense to me. So that's ideal. I think it'll be really useful.
I'm glad to hear that. So yeah. By the way, Vadim is here, so he is writing all the small functions and features, and still adding a little bit more like flexibility.
Vadim. Okay, yeah. So
Vadim.
awesome.
Vadim's
Thanks for doing that, man. Like, you don't need my help. I think Vladimir can help you with everything. But if you need anything, let me know, and I'll be happy to try to help. So I have a question for you. I've worked with your writing a little bit now, and you're a good writer. In Russian, is there does the exist, like the article the?
We don't have articles at all.
There we okay.
That's why we do not use them in English.
Yeah, now I understand.
Well, I mean, try to use them, but it is always like you need to like enforce yourself, and so we don't have this kind of intuition how to use them. Yeah. Actually, also even like in English and in French, they use in a slightly different manner.
That's true. Yes. But those map onto each other pretty well. Like I basically understand how to make French English work together. I just noticed this because you're like I said, your writing's very clear, but every now and again you read a sentence like, wait a second, there's something missing here. I need to go back and. But I'm good at that, so no worries.
Cool. Okay, I think we are ready to begin our last lecture, and yeah, today we have again Russell Corbett-Detek from University of California. So, Russ.
Sure, I'll take it away. Yeah, thank you. Okay, so today I will just go through some of the last and most exciting stuff that I think is going on right now. I can't share until you stop, but the basically what I'll be talking about is some of the work that we've seen. First of all, I'll revisit a few topics from last time and try to clarify a few things. Then I will revisit some of the work that's been going on in detecting and understanding natural selection in the SARS-CoV-2 genome, so both biology of why, and then a little bit about the approaches people are using. And I think, I'll speculate about what I think the most promising ways to do this are going to be. And then I'll talk a little bit about our work and other work on trying to detect recombination in SARS-CoV-2. There is recombination. It's not super common, but it certainly exists. So you've actually disabled screen sharing, by the way, Vladimir. So I need you to enable me if possible.
I think you should be able to share already.
It says
no, sorry.
No worries.
No, you can.
Okay. Very good. So, and I'll warn you in advance. I remember last time I left you with some homework, which was to figure out how to run that SnakeMake module I'd given you. So we're going to do that today, and hopefully it'll work. If not, you're going to watch me do it, and you'll see how it works. So, okay, over here somewhere, I feel like it might be these. Yeah. Okay. Very good. Present. So, okay. Like I said, tell you today about both recombination and a little bit about natural selection in SARS-CoV-2 and kind of what we're doing with these things and what I think the future is going to be. Like the vast majority of problems in phylogenetics and evolutionary genomics, the data set we've collected is just so different, right? It's a ton of incredibly closely related samples. So the vast majority of existing methods are actually they perform kind of poorly in some cases, and that's certainly been true for most of this stuff as well. And so I think it needs a big rethinking about how we do a lot of the stuff we do, and I'll tell you about that. So as you may recall, last time I showed you that maximum parsimony and sort of extensive optimization based on maximum parsimony is almost certainly substantially better than maximum likelihood approaches for these sorts of things. And that's really, I think, counterintuitive to a lot of us. And I think it sort of harkens back to this bigger idea that these data are so different. The way we're collecting them is so different that we need to think pretty critically about how to reinvent the way we do some of the stuff to meet those data where they are. Okay, so that's today. Briefly, I want to revisit this because I feel like I can show you a more compelling image of why this is a problem before we go on too far. So as I mentioned, one of the core issues that we encountered really early on in this problem was that the way the data are collected, a typical genomics project would be one or a handful of sequencing centers process all of the samples, right? So they all go to a few places, we do them all. And so you'd have at most 3 or 4 batch effects at that point, right? Or at least batch effects to the extent that one sequencing center does something unusual. It should be obvious because it'll be one of 3 or 4. In SARS-CoV-2, what's happened is that there are thousands of sequencing centers. They're all over the world. They all have extremely varied experience and skills in doing this kind of thing. And so, and of course, it's hard as well. And so lots of little things don't work right, and they're unique to each of these places. And so what that will do is it will introduce these kinds of systematic errors or mutations that we see. So if your true phylogeny looks like this, where there's exactly 2 real mutations here and here, but we have these recurrent errors. In other words, they're systematic errors that pop up in lots of different sequences. Well, those shouldn't really be correlated with the phylogeny. And in fact, they're not. But because we don't know that, when we go to infer the phylogenetic tree, there's a good chance it will affect the way we group some samples. So for example, sample 6 here has now been grouped into this clade with 7, 8, and 9 instead of where it really goes with 4, 5, and 6. So this was a tremendously important problem. I think we all recognized it pretty early, had a pretty good idea this needed to be dealt with, and started working on it quickly. And I think I showed you this last time, but this is kind of what this can look like, which is essentially the parsimony score is the number of times the mutation is inferred to have occurred on my tree. And this is my alternate allele count, where of course, the more times I see a mutation in general, probably the more times it will occur as well. There should be a relationship between these things. But we don't necessarily expect this nice, strong linear relationship here. And so that's been the thing we focused on to identify the strongest outliers in this whole thing. So before I go on to the next bit of review, I want to show you, hopefully you're all seeing this now. This is Taxodium. It's a viewer, and I'll actually show you how to get here too, even though it's kind of obvious. You can follow along if you like on your web browser. If I do this, view public tree, what that's doing, you can actually see the link up here. It's pulling from our server, hgwdev.gi.ucsc.edu. It's actually Angie's development server. So this extremely professional looking URL up here is because it's Angie's stuff that we're loading up. But anyway, you can get into it pretty quickly. And this is, to our best approximation, the current public SARS-CoV-2 genomes. So it's 2.25 million sequences. Pretty well understood at this point how this works, but basically this is your alpha here, this teal thing, B117. This is delta, which arose from India and then spread pretty quickly through most of the world. This is AY4. You've probably heard a little bit about AY4.2 recently, right? So you can scroll in and out on this very quickly. And if you look down here, you'll see, yes, these are the mutations that we're looking at, which is 145H. We don't know if or why that's important, but it may be, and 222V. They're both in the N-terminal domain of the spike protein, which, by the way, it's showing it right in the center there where I'm hovering over the node. It says S:A222V. That's how you can see the amino acid substitution here. So we don't know if or why these are important necessarily, but we think they might be. And so that's what we're trying to work on and deal with right now, is to figure out how to make sense of this new lineage that does appear to be spreading pretty quickly in the United Kingdom. It's not really in the USA yet. I don't think it's in Russia, but the data from Russia is not super up to date, so it's hard to know for sure. Or at least the data I have from Russia is not super up to date. And so that's kind of that. That's how you get oriented to the tree. The thing I wanted to show you is, what does a systematic error look like? So if I do this and I say amino acid substitution, there's one that I know is a systematic error, and it's associated with a very specific sequencing technology, which is the V4 primer set from Arctic. So if we go to ORF1AB, I'm pretty sure it's this, yeah. What this is doing now is it's highlighting all of the inferred occurrences of a mutation at this position in the genome, 5086 ORF1AB. So you can look at this and see there's something that doesn't look right already, right? There's this imbalance in the tree where everything up here, almost none of them even have this mutation. I think there's exactly this one right here in B117, And maybe there's one there, not sure. Nope, that's actually down in alpha, in delta. So B117 and then down in delta is where it's all happening. And this would make sense if the error was relatively recent or interacted with the genetic background of delta. In this case, it's because the primer scheme has just been updated and all of the new samples are down here in delta. So of course it's in delta. What you can see also is that there's even an imbalance within delta, right? If I zoom in a little bit, what you can quickly see out of this is that, okay, AY.4, this brown thing down here, has an absolute ton of these mutations, right? It's all over this thing. These others up here have much less. And the reason for that is there are things like AY39 and AY25, which are mostly in the United States, and B117, this at least clade here is mostly in the United States. AY.33 up here as well. But of course, if you zoom in a little more and you see these, oh, those are back in England. So what's actually happening here is that COG UK has an incredibly high error rate at this particular site, like insanely high. And so that's, this is what we're seeing coming online right now. It's there's another one as well, but I won't go into it. But basically that's what this can look like, is that you have a particular mutation that is occurring all over my tree and it's very strongly associated with one particular group. That's a systematic error. In the case of COG UK, it's easy to see it because they make, you know, about half of all the public sequence data. So it's not very hard for me to find this stuff. But of course, you can see them in much smaller scales as well in some cases. Okay, that was that. Hopefully that made a little bit more sense. Again, that's taxonium or taxodium.org. So highly recommend you check it out. I'll put it in the, can I get to the chat? I don't know how to do this. Here we go. If you check that out, you can play with it. I really enjoy using this viewer. I find it incredibly helpful when I'm trying to figure out what the heck is wrong with my tree, because there just aren't, as far as I know, any other useful approaches for viewing a tree on this scale. So you really need something like this. Okay, so let's return to where we're at. Moving on from there, the other thing we talked about that we didn't really get to do the example of, and I'm hoping you all got your Conda install working. If not, well, this exercise is also not going to work. But just to remind you what we were talking about and why I think it's important. So what the vast majority of public health applications are trying to do in genomic epidemiology is this. They're trying to find how many introductions there were into a given area and a group of infections that are descended from it. So how big the cluster got. So a cluster would be the number of samples that descend from that introduction. And the introduction event is this branch here, presumably. Okay. And again, some design constraints on this, right? So the big problem with this has been that even 1,000 samples can be really problematic when you run BEAST. And so there isn't, and so people often find kind of hacky ways around that, like they run BEAST, but they keep the tree constant, which sort of defeats the entire purpose of BEAST as far as I'm concerned, and other things like this. So they don't really have a good solution, and it doesn't scale at all, right? Like you can make in a day, big sequencing labs are making 10,000 samples. That's way more than you can run in BEAST ever. And it's certainly more than you can run in years. And so it doesn't, our ability to use these data is really stymied by our analysis. And so what we really need are simple things that can be done very, very quickly and are good enough to give us most of the answer. And so our design constraints are, we need something that can be run daily, right? I want to be able to run this in 20 minutes every day on millions and millions of samples. You have to be able to handle polytomies. So you got trees that have this crap going on where there's a ton of literally identical samples. Got to be able to handle that. Oh, sorry, I need to be presenting. I'm just talking. Okay, you got to be able to handle polytomies. And you would like to have a portal that has simple and intuitive data exploration platform. And I won't talk too much about that, but I think we have a way that's pretty good for this. What we came up with was this heuristic. So just to remind you, the way this worked was you basically define the states of your samples. So they're in or out of a region. Really, the way we do this is we define a set of regions. So this would be region 1. This might be in region 2. There's others in region 3, et cetera. And all we're doing is a single traversal of the tree. So post-ordered traversal, which typically means you start down at the tips and you go back up. So if we start here, we say, okay, well, both of you are in the region. So clearly my confidence that you're in the region is very high. So for this node, given that all of its descendants are in the region, I think there's a pretty good chance it's also in this region. You go up to this one, the same is true, right? All 3 of these are in the region. We're going to call it as in. You go up to here, and now you have this choice to make. And the way we ended up doing this was we wanted to weight by both the genetic distance to the nearest sample. In other words, how close the nearest representative from a given descendant branch was to be in the region. So that would be the number of mutations, so 1 in this case, and by the number of samples, right? You're gonna have a lot more confidence because of inference errors and all sorts of other things. You have a lot more confidence that you've found something real if a bunch of samples from a region are associated with a given cluster, right? So we have 1 mutation, 1 sample. In this case, the genetic distance is 4, right? There's 3 mutations on this branch and 1 mutation on this branch. So 4 mutations, 3 samples, we get this other ratio, 1.33. And so again, we just throw it under a squash function to get our confidence in this node. In this case, it dips below 0.5, and we say, okay, we're going to call this branch as the introduction into the red region. And we might say that, okay, it's possible that you came from this blue branch, but we may not know that. Okay, cool. So I'm gonna, again, this is a test if we did our homework. So go try to do this. Hopefully you've got your Conda and SnakeMake working now. If you don't, I'll come back and try to just show you how you would do those things really quickly. But I'm also gonna upload a slightly different file. So it's not the exact same file as last time. So we can have a little bit of fun with this. 1 second, we'll set you off on that while I get more coffee here. Okay, where was Sorry, I have this. Workflows. All right, and that's going to take just a second to upload to the chat here. You can grab the file from there and try to do this thing if you can. So that should be up. The entire set of commands you should need to do this, provided you have SnakeMake and Conda installed, is right here. So give it a shot, see what happens. I will be right back. Okay. This time, who got it?
Well, I didn't get it because I forgot my Mac on my job.
Oh my gosh, you're killing me. Okay.
So I can miss it in front of this, but I have a theoretical question, and maybe it will give other people a couple of extra minutes. I was just curious about the clusterization algorithm. Were you able to produce, using this algorithm, results you didn't like on artificial trees? And if you did, what kind of trees are they and how do they look?
Yeah, so that's a really excellent question. We are still in the process of shoring that up. Is why I don't have nice figures to show anybody about this. But we obviously did run this on simulated trees. So in fact, we used VGSim to simulate our trees. And so what we did in that case was a simple 2-region model we varied the rates across different simulations of migration between the regions. And we just said, okay, 2-region model, and then sampled 1 million sequences and threw down mutations approximately equal to the number of mutations you see in SARS-CoV-2. The tree structure is not exactly the same, of course, because there's a billion other things that affect that tree. So I won't claim it's exactly like reality, but at least has a similar number of mutations per branch as in real SARS-CoV-2 trees. So that's kind of how we got our starting tree. And then what we did with that tree, so that was VGSim into FastSim, right? Which is the software packages that, well, you guys wrote VGSim, and then FastSim is our friend Nicola, who wrote the sort of package to generate variation very efficiently. Okay, so we did all this, and then we threw this into MatUtils introduce to figure out, okay, do we actually do a good job or not? And the simplest way to evaluate that is what's called an adjusted RAND index. RAND is R-A-N-D, which is a way of evaluating how similar 2 sets of clusters are. Right? So if you have a bunch of samples that are in a region and you cluster them, you can evaluate how similar your clusters are. And the adjusted RAND index is nice because it adjusts for chance. In other words, at some rate, you must throw 2 samples into the same cluster by chance and get it right, even though that had nothing to do with your algorithm being any good. So when we do this, for parameters that are at least somewhat like SARS-CoV-2, we end up getting an adjusted RAND index of 0.92, which is pretty darn good. The other thing we've done, so that's simulated data, which is your question. The other thing we've done is take much smaller data sets people have analyzed for the pandemic. There's been a few where they estimate the number of introductions and the size of those introductions into a given region. And so we've taken those and we've done exactly that same analysis, only we're replicating what they did with maximum likelihood or with Beast, and we're doing it in our software. And when we do that, the answer tends to be, we get almost the exact same number of clusters, the same clusters in general pop out. So it typically works very well. The places it doesn't work as well are really, really, really big clusters get broken up. I don't know exactly why this is true, but they tend to get broken up into smaller clusters in some cases. I would argue that probably isn't the most important thing, because the whole point of this is to find the clusters that are like right now, which tend to be pretty small and growing slightly. So that's what we're looking for. Does that answer your question a little bit?
Yeah, sure. I assume that these very big clusters were kind of deep ones, not like one parent and 1,000 children. So that's like a common problem with any clusterization, but you just have to stop somewhere at your definition of what actually cluster is. And since you can't formalize it, well, there is no way for your algorithm to satisfy every wish of yours.
That's 100% correct. This is a tool for exploring the data. I'm not saying it's a finalized analysis, right? Sure. Like this is just for public health to say what's happening in my region right now, and that seems to be useful, I think. And I can show you guys how you would evaluate one of these, or how I've had people evaluate these in the past, real quick, which is just to go. So what should have come out if you were able to run this, and I'll show you what happens when I run it. Actually, one other cool thing about this, it doesn't say this, it's an unknown feature for most people. So this is the file that should have produced, oh no, sorry, you can't see that different screen. This is the file it should have produced, user-seq-texodium.pb. So it also uses what's called a protocol buffer. If I just drag it on here, it'll load. And so what the SnakeMake workflow did was it downloaded the latest public tree, which we distribute as this kind of file, a mutation annotated tree. It added the sequences to the tree, and then it makes this visualization by using the same metadata file, but with your sequences added. So in this case, I need to get a sequence ID so I actually know which ones I'm looking for. So give me 1 second. But the biggest cluster you should have found was one that was about 45 samples large, or approximately 45 samples large, and it had a name. We go to sequence name, search for this. There are 2, which is more than I would like. There should be 1. So let me see if I can make that more specific somehow and not have that problem. So I just grabbed 1 here. Let's try that. How about that? There we go. 1 result. So if we go to locate, here we are. We're in this cluster. They're all gray when you color by lineage, and that's because we didn't tell it the lineage, right? I only have metadata for the public samples, so these were just added. I don't know. But you can pretty clearly see the lineage must be AY30 because everything around it on the tree, if I hover over it, shows you AY30. In this case, this is the biggest cluster that we called. It's 45 total samples out to here. It is possible, of course, that these 2 are also part of the cluster. That's where this algorithm is tricky, right? Is how do I decide what I should merge versus not? In this particular case, they don't get merged, and it's because there's other descendants of this node that are not in this region in Cambodia. They're in California. I don't know what that means, but that's what we got. In this case, if I asked you where they may have come from, you would just sort of follow up the tree, and you would see, okay, well, these are all in Thailand, and these are all in Thailand, and there's a few in England and Switzerland, but the vast majority are in Thailand. So this cluster's in Cambodia. I think it's reasonable to suppose this probably was introduced from Thailand. And in fact, if you are able to produce the output of this run, you'll see that it's pretty darn confident that this cluster came from Thailand as well. So the point of this is just to say, because the data are so dense, and because we are not struggling to produce enough genome sequences, a lot of the approaches people have come up with in the past for these sorts of problems are about dealing with the uncertainty of having very few samples in a much larger pandemic. What's happening now is that we sample, in some places as much as 100%, but pretty commonly as much as 2 to 5% of all samples in a region, which means that there isn't as much uncertainty. Like, I've got a pretty good idea what variation is present in Cambodia. I don't need to do anything really fancy to figure this out. And so that tends to be the solution we're working with here. So okay, with that, hopefully that made some sense. I would like, if you guys get a chance, try to play with this thing. It can do a lot more than just introduce and taxodium. So if you ever have to analyze some sequence data for SARS-CoV-2, this is a really, really quick way to do it. I maintain a very large, very well optimized tree for you. So it should be much easier than making your own tree with a much smaller subset of the data. So I highly recommend this. Obviously, I'm convinced this is the way to do this stuff. So give it a shot. If you get a chance, let me know if it worked for you, etc. And then the answers to the questions that we would have asked were, there are 5 clusters. I showed you the big one. There's also one of size 12, 9, 2, and 2. The largest one probably came from Thailand. Okay, so with that, let's return to the main topics of the day. So a little bit about selection and a little bit about recombination and how I think we're going to go about studying these things in the future. And some of you are working on selection, so maybe you'll chime in and tell me what you're doing exactly, because I'm very excited to hear about it. But anyway, I'll tell you what I know about it too. Some of it anyways. So again, this is your SARS-CoV-2 genome. I introduced this on the first day and probably again on the second day. So hopefully it's getting pretty familiar. Your gene structure is along the bottom here. And the gene that everyone's going to talk about in the context of selection is right here. It's the spike protein. It's this protein. It's the kind of receptor binding chunk of spike in particular that seems to be the most important for entering host cells, which is sort of the key thing that you need to be able to do if you're going to replicate a virus gene. Okay. This is going to become relevant mostly in the context of the recombination section. And I'll just emphasize the one feature that there's a lot more RNA because of subgenomic RNAs. There's a lot more RNAs down here in the back part of the genome because you have very, very small ORFs. The recombination function that produces these subgenomic RNAs recombines these translation regulation sequences with each of these starting points and translates from there to the end. And so again, you see these big proteins, long tails, poly A tail because it's eukaryotic translation. and you have a lot more RNA in the back part of the genome. That's exactly what I just showed you there. You get this recombination canonical kind of thing where you want to get this TRS-L sequence associated with each of these transcripts, or if 1AB is translated as one transcript from one transcript with a ribosomal slippage point right here, the others are all sort of separate, so they're all their own little subgenomic RNAs. And this is what I was talking about. So you get a lot more of the RNA abundance sort of to the back chunk of the genome. I won't talk about it a whole lot more than that, other than to say that it is this is tremendously important for recombination when I get there. But I'm actually going to do this in reverse. I'm going to talk about selection first. So, okay, there we go. So one thing that I think I pointed out probably way too many times, you guys are getting bored of this, but it is really important, is if you look at this plot, and this is a plot of the similarity, in other words, nucleotide identity between a bunch of different genomes in SARS-CoV-2. So many more have been sampled since then. There's an entire research program that's essentially based on going out into caves in Asia, especially Southeast Asia, grabbing random bats, taking a blood draw and sequencing whatever's in that blood draw. So I'm not kidding. This is literally what a ton of people are doing right now because they're trying to find closer and closer relatives of SARS-CoV-2. And in fact, the closest one that's been found so far is this one here, BANAL 20-52, this yellowish line is kind of along the top here. You can't see it in this part, but you can really see it in spike here where it's very, very similar. The other thing I'd like to point out is there's this enormous excess. So in all of these comparisons, you see much more nucleotide diversity right on top of the spike protein, and in particular, right on top of the receptor binding domain, which is kind of right here down the center of this thing. So we think this is going to be a really important point of adaptation for the virus, and I'll talk about just why right now. The basic reason we think this is true is that it's a mutational arms race, right? So the vast majority of host pathogen interactions are this kind of arms race where one is evolving in order to subvert the genetic defenses of the other, and then the other is evolving to suppress the spread of the pathogen, right? So if my virus is this and my host cell receptors are this, and they need to correctly bind the cell receptor in order to enter the cell, this little interface here, right, the spike protein receptor binding domain, and then the human receptor, ACE2, is going to be a major, major site of evolutionary innovation and evolutionary conflict, right? Because again, the virus needs to beat you, and so it has to be able to enter. And so over very long periods of time, we expect this sort of dynamic to occur where the receptor may mutate to avoid infection. And so these, if this is our sort of lock and key allegory, you see these things match exactly, right? They have these nice curved features. This matches this curve, so it does really well. Over time, the host evolves away from that. And then, of course, the spike mutates to reinfect. For what's happening on the very, very short time scale within humans, probably the host receptors are not evolving because, our generation time is way too small for that to be the big difference here. And so this is sort of a longer time concept. In shorter time, I think it's much more likely that what's happened here is that the virus actually enters human populations something like this, right? Because it wasn't already in a human population. It hasn't had lots of coevolutionary history with humans to evolve an exact binding match to our ACE2 receptor regions. And so what it does is it's going to evolve to this sort of thing where it says, okay, the ACE2 receptor will evolve to be a better fit, or sorry, the spike protein evolves to be a better fit for the ACE2 receptor under the sort of lock and key allegory for what's going on here. I'm sure it's much more complex at the actual molecular level than this little cartoon, but the basic concept is this is a major site of evolutionary innovation. We kind of expect, I'm sure other sites in the viral genome matter quite a bit, but this is the main thing that I expect to see affect SARS-CoV-2. So, okay. And just to remind you, we won't go into great detail on this. This is something from the first day, which is there's experiments we've done that kind of told us a little bit about what was going to happen in the future of SARS-CoV-2, where you could take all possible mutants of spike proteins, or most possible mutants of spike proteins, express them on the membrane of yeast and measure binding experimentally. And then you'd measure this whole library and sequence it at the end, and you're basically looking for the places that affect binding the most. And Of course, in the vast majority of places, binding gets worse because most proteins are pretty good in the first place. But of course, there's a few cases where it actually gets better. And one of those was probably D614G. There's been others as well that I'll show you in just a second, because I really like these videos and I think they're kind of evocative of what really happened, which was incredible waves of adaptation of the virus. So, just to put a very fine point on this, viral mutations that increase transmission must become more common over time. So if we imagine this is sort of patient zero of this new mutation, and they transmit one copy with the ancestral form, the red variant, and one with the blue. If the red is pretty transmissible, right, it transmits to 2 or 1 people every time, it grows for sure. But blue transmits to 3 or 4 people every time, and of course grows much more quickly. And so over just a few generations, it can become the dominant form of the virus in a given area, because of exponential growth, because it's compounding, right? If it's 3 and then and then down here at this generation, I now have 10. It goes really, really quickly up. Okay. Hopefully that's fairly obvious to people. We can talk about the details of that if you like. The important point is that it's 2 competing exponential growth processes. So it's really the ratio of those growth rates that's probably the most important factor. Of course, it is possible the generation time gets smaller, but I think it's unlikely. So, okay. I think I may have shown this the first day. The D614 sweep comes pretty quickly in the origin of the pandemic. So this is one of the beautiful things that we got from all these viral genomes, is the ability to watch this happen in, it's not real time, it's weeks delayed, but we do have a pretty good idea what was happening as we're going. So at the beginning here, you see this spread pretty quickly, and then the mutation arises in blue, basically, bluish green, and it quickly spreads throughout the world such that it becomes the most common type kind of everywhere very quickly. And in fact, D614G is in every single virus that I think we sequenced today by a pretty good, and I don't think there's any others. So it's really gotten out very quickly, and it's spread very fast. That was sort of before we were even paying attention to variants, because most places were still spinning up their sequencing programs and didn't have quite as much going on. So the more interesting ones came along a little later. So alpha, for example, is one of these where it's going to show up just a second here in orange, I believe. Where it spread from the UK very quickly out into the rest of the world, and just, it swept through everything. Everything turns orange all at once, right? It's really, really fast. That one's quick. It's a very fast process. And so the idea there is that again, at this time, we actually had pretty good viral surveillance up and running by this point, at least in the UK and most of Europe. And so we were able to see this extremely quickly. Okay. So, damn it, I'm having a lot of trouble with this thing today. So, okay, and the same thing again, oh my gosh, happens again with Delta, only it's even more extreme, and we had a ton more sequencing, right? So these circles are proportional to the number of samples. You see they've been getting bigger through the pandemic. So we had this first sweep with D614G, and then Alpha, and now Delta. And Delta is a hard sweep as far as I can tell, in the sense that every single virus that's sequenced today is Delta. And so we have other stuff in gray and blue or whatever. What's going to happen here in just a minute is green will pop up there. You can see it's coming in India just a little bit, spreading out, and then, come on, it will start elsewhere in the world. And it's this incredible, there we go, this very fast process once it gets going of everything just turns green. And the time scale we're looking on here is like 2 months or something like that. This little bit of holdout here in yellow and red is Mu pretty much, and a few other South American lineages. It looks like they're also not going to win against Delta. So Delta is the most transmissible thing, and just about everything sequenced today is Delta. So again, the idea is that selection has been a really important part of determining what happens to this virus. And there's been so far, kind of repeated waves of adaptation of the virus. So D614G was basically a hard sweep that happened very quickly before we were even really watching. And then there was what looked like partial sweeps of alpha, gamma, beta, things like this. And then Delta came along and did another hard sweep. So the assumption is that alpha was about 50% more transmissible than the ancestral form it displaced. And then came along Delta, which was again about 50% more transmissible than alpha and other viruses circulating at the time. Those numbers have enormous confidence intervals, so don't quote me on 50%, it's a number. a large enough number that selection is very, very strong. And as soon as it got going, you see it just took over very quickly. Okay, and this is doing it again. Come on. I really shouldn't put videos and slides. It's always a problem. So other evidence for selection is things like this, where we see that there are actually many independent mutations of the same allele. So what's going on here is that at least at the beginning of this, we saw this one mutation, N501Y, pop up several different times, right? It was in alpha, it was in beta, it was in gamma. And you can see it's on very different branches of the phylogeny, here, here, and here. And so this is pretty good evidence. Sorry, in and of itself, it's not very good evidence anything, right? A mutation occurring 3 times is actually very common. But if those mutations occur 3 times and you see the lineage grow quite a bit immediately afterwards, I think it's pretty good evidence that this is something that's adapted for the virus. Of course, Delta does not actually have 501Y. Delta has other stuff going on. But the important point is that you can see these kinds of things happening. And this is one of the ways, I'll show you in a second, one of the ways people are thinking about detecting and understanding natural selection in virus genomes. What are we at here? Okay, so how do we do this? How are we going to estimate this, right? So the first approaches that were used were like Eric Voles and others were working with sort of one variant versus the field. In other words, this is all data from alpha, where we're basically comparing this one lineage, B.1.1.7, against everything else, which sounds good, except that I'll just tell you right now, I don't think it's really appropriate for SARS-CoV-2, or it hasn't been until recently, because there were actually many, many competing partially selected lineages all over the place. This kind of comparison probably wasn't all that reasonable. It certainly isn't reasonable for most cases, although it may have been for this specific one looked at here. But so obviously, if a variant is sweeping, there's a good chance it's more transmissible. What they showed was the estimated frequency of what they called a VOC, a variant of concern, B117, which is alpha, across several different places in the UK, right? And so you can see that it pretty much spread through everything at a relatively similar rate. Like these lines have extremely similar curves to them, suggesting it's done sort of the same thing in each of these places, which is great. It tells us that there is information. It tells us that we're learning something about natural selection in this case, or potentially we are. So, and so what you can do, there's a couple different approaches. This one is one of the more model-based, where they tried to estimate using these the effective population size of each of the lineages through time. The idea being that the ratio of these two things told us something about the relative growth rate, because of course, if your population is growing through time, the relative rate must be sort of encoded in this. The trickiness is that there's a ton of other epidemiological factors that affect the spread of the virus. And so you really want to have these comparisons, right? So I'm comparing B117 with B.1.177, which I appreciate is a confusing set of variants to compare. But the idea being that this one was actually quite common at the time, B.1.177. And you can see that it didn't really grow, right? It was pretty flat. In fact, it tapered off a little, whereas alpha, really had this nice exponential growth to it. And so you can use the relative rates here to say, okay, that's the relative transmission advantage that this has if we estimate this as an additive thing. And if you do this, you'll end up with a number like 0.5. The problem with these approaches are they end up subsampling to just a few thousand genomes, which means you throw out the vast majority of your data, and you're really only able to compare 2 lineages. So it's just not really the right way to be doing this, in my opinion, because it's computationally inefficient, and it's just not going to hold up to the flood of data that's coming into this process right now. So, and the other main question that I alluded to a little bit at the beginning, and hopefully is sort of obvious why this is important, is what should we do when there are tons of selected variants simultaneously, right? So probably it wasn't just alpha that was spreading through the world. Maybe in England it was, but we saw other major variant lineages kind of come up and spread. And so it strongly suggests that we had many, many of these things going on all at once. One thing that we can use, a number of people have noticed this, and I think is probably the most promising thing, and it's kind of what I'm hinting at back here, this slide, is to say that if we see the same mutation arise many times and it's associated with a growth rate many times relative to its sister lineage, that tells us something about how much that particular mutation might matter. And if it happens enough times, you sort of have it averaged across many different genetic backgrounds, which is pretty cool. And so this is just a figure from Lucy Van Dorp, which all my references are cut off on these slides, but I promise you it's Lucy Van Dorp's figure, where they were looking at the, early on, just looking at how frequently it looked like mutations had occurred independently of each other along the genome. And so you see a couple of places on the genome here where there's just an absolute ton of the same mutation occurring independently, right? So this is 10. There's 5 or 6 that occurred at least 10 times in just a few hundred genomes. This particular position here is sort of infamous. It's 11083. It flips back and forth all the time on the tree. And so it does not have very strong evolutionary signal. And of course, there's many others as well that are also highly recurrent positions in the genome. So if we could use that, what could we do? So the approach these people came up with, which I do like and I think is generally reasonable, was to look at the evolution of this thing they called the ratio of homoplastic offspring. The idea being that, like, if we go to this node here, 2, so ignore this stuff for now, it's just a figure grabbed from their paper. If we go to this node here, 2, and we say, okay, here's a new variant allele. Obviously, I don't sample these internal nodes, I only sample these descendant samples. If there's 4 samples here and 2 here, we might say that this has grown in total, cumulatively across the entire time to be twice as large as this. That's cumulative, right? It's obviously not a single time slice which is how you would really do this kind of comparison, but that's still a useful way to think about this. And they came up with a bunch of rules to filter these down. I won't go into great detail about that because I don't think it's all that interesting, but you can read their paper, learn about how this was thought to work at the time, and get an idea. But the basic idea is that this node 2 is not unique. There are probably many, many nodes in the tree where the same homoplastic allele arises. And if you see this bias where every time that mutation arises, or most times that mutation arises, it grows to a much larger total size, this is a really nice way to look at these kinds of mutations because you see they should be more or less averaged across the various genetic backgrounds of the virus. But more importantly, they also have the same sort of local epidemiological factors, including like sampling rates and whatever the local growth conditions are, right? So if this mutation arises during a lockdown in this particular case, well, the nice thing is they're both, this set of descendants and this set of descendants are probably in the exact same lockdown. So they have the exact same, for a given occurrence of a mutation, that plus its sister node have the exact same epidemiological parameters going on. Okay, so that's a tremendously useful observation. And so what you can do then is basically say, okay, if we compute this ratio for every single one of these and then took the mean, what will we find? And so they did this. This is log 10 of this ratio, right? So if you log a ratio, it can be very nicely behaved. And that was the idea here. And then we looked for positions that seem to be occurring a lot. And this is actually fairly early on, right? So this was about 10,000 genomes at the time, such that, okay, we didn't see a whole lot of really strong effects. And in fact, some of the biggest effects they observed that look like negative selection were actually recurrent errors. They've since been removed from the figure, but at the time, there were quite a few recurrent errors in here, and this was problematic. But the important point is, yeah, you see this sort of same basic pattern of, well, these all sit around 1, which would mean the vast majority, or 0, which would mean the vast majority of these are not strongly selected. That is, the descendant clade size is almost the same in the 2 different approaches, so it's not particularly different. This is cut off like crazy. So you see an equation, but there should be more. Shit. Okay, I'm just going to skip that and talk about it just verbally. So the way this works is you obviously might want to expand on this approach a little bit. And so the way that they went about doing this was to say, well, let's solve for, let's use a much bigger set of data and use a very similar approach. Now it's called SEGA, which is the coefficient of exponential growth alteration. It's the same basic group. This paper is from Damien Richard and Francois Ballou and Lucy Van Dorp, and many others, of course. In fact, I'm on the most current version because I helped a little bit, although I don't think they used what I did in the end, but that's okay. So anyway, the point is, we were able to revisit this thing, but now we're using about 700,000 genomes or 600,000 genomes. So you have quite a bit more information to work with. And there's some pretty interesting things that start to emerge from this pattern, right? So as opposed to before, where there really wasn't strong evidence for selection on any one site that we could really get out of this, with Sega, we're actually able to do a little bit better, which is to say that because there's so much more information here, we see each mutation so many more times, we can find some real outliers on here. So if they all sat at 0, it would suggest that every mutation was about equally likely to negatively impact or positively impact growth, right? They're all sort of neutral. There's actually a negative skew, which I find pretty interesting. So across all the sites, this is using both the mean and the median Sega in a given site. They're both negatively skewed, about 0.48. So you can see the line right there and then the line right here. Okay, so this I find tremendously interesting in the first place. The second thing you can do, though, is we say, okay, well, let's just take all of these semi-independent estimates for each position along the genome and try to estimate an additive transmissibility. That is, essentially add up this effect on the growth rate across a bunch of different samples through time and plot this. What you see is something like this, where initially the growth rate was certainly above 1. It actually did a little bit of adapting very quickly, descended slightly, and then of course alpha arose and we were up here for a while, and then of course delta arises again. And one thing that's actually a little bit hard to see from this figure, but is also tremendously interesting, is that each of these major bursts of adaptation gives you a real big jump, right? You jump up each time in total transmissibility, or we don't know that it's transmissibility, but it's likely to be transmissibility of the virus. But then you see that as mutations accumulate over time, most of those probably negatively impact transmissibility. You see it start going back down again. And each of these it is actually happening in each of these cases, right? You have this little burst here with D614G, and it goes back down. The same is true here for alpha, which starts strong and then goes back down, and then again for delta. So we think basically that the sort of tells you about the tempo and mode of adaptation in the virus, where big adaptive events may not be super common to see these brand new, very strong lineages coming online. But of course, over time, you should accumulate weakly deleterious mutations that decrease your transmissibility through time, right? So this is sort of this ongoing struggle that a virus will face, which hopefully is not too controversial. It kind of makes sense to me that this would be true. I think there are lots of important components to the way this is estimated that make it a little hard to know for sure this is what's happening, because the number one issue is that there's very, very strong circularity, right? So if I see that a particular mutation grows to a very large size, and then I go back and I estimate the transmissibility of delta based on the mutations that I discovered in delta that grew to very large sizes in delta, then I've got a problem, right? Because I'm using the mutations present in the sample to estimate the transmissibility of the sample. And so I think the challenge of this is to do kind of a hold one back or something else, where we don't use delta. We just say we've learned everything from the first part of this. Can we predict delta? And I don't think it's done that well so far, which is kind of an interesting problem. And I think what I mostly want to highlight here is not that this is a solved problem. It's clearly not. Is to say that forecasting, so using the information we have right now to forecast the transmissibility of new lineages, is tremendously important. It's also incredibly hard. I think we have been, incredibly bad is a nice way to say this, incredibly bad at using a genome sequence to estimate the future transmissibility. And I've done this myself, and I think many others have as well, where we think, oh, that has important mutations, so it will be an important genome. The only thing that's really been useful so far has been how much it grows over how much time. And this is one of the ways starting to get toward forecasting, right? If you have this list of positions that we think have certain effects on transmissibility, then you can sort of forecast the next one, but I think there'll be a lot more to be done there. And this is one of the ways I've seen that's come online to do this is this really cool paper from the Broad. So Danny Park and Jacob Lemieux and a few others did this. But basically the idea was, okay, we'll take millions of samples out of USAID and we'll make an alignment, and we'll call the lineages to whatever extent pangolin actually works. And I won't go into detail about that, but we'll call the lineages, and then we'll get we'll look at those lineages across time. And so we'll have these sort of nice frequencies across time and space. We also know which mutations are present in each lineage, so we call sort of a consensus of the lineage. So they're trying to do this in a phylogeny agnostic way, where we just say, well, it's not totally agnostic because pangolin is a phylogenetic approach, but but we're trying to do something that doesn't require me to infer a tree of 2 million samples, which is what I did, but they did something else. And so they do this sort of thing. And then they have this, I won't go into detail about this because I don't actually understand the whole thing completely, but basically it's a machine learning framework for trying to figure out which of these things is most likely to contribute to growth. And the idea is that we'll get, at the end of this, we'll forecast the per lineage growth rates, extract mutation-specific coefficients. So in other words, we'll deconvolute the lineages into the shared mutations and just get the specific mutation-specific coefficients and then make predictions from there. These are still completely additive as well, and I'll talk about why I think that's a concern in a minute, but just to show you what you get when you do this, well, this is growth over the ancestral growth rate. So essentially it's the increase in transmissibility over time, and of course, as you would expect, it goes up. You see this really nice curve here. There's some interesting stuff that, and this is, the x-axis here is just the first observation of a given pangolin lineage, so the day or the date the first one was observed. So that does mean that there's sort of a type of ascertainment bias because in highly sampled regions like England, you probably see the first one a lot earlier. So B17 over here is actually, it was, they're plotting this a little funny. It was back in September. I don't know why it's over here in December, but the important point is you get these really nice, you have this nice kind of relationship where of course the virus is increasing in fitness, and we can use this to forecast this. And it does appear to work pretty well. So for example, they did the thing where they trained the model leaving out alpha, and then they predicted the growth of alpha at the time, and they did quite well as far as I can tell. And they're trying to do the same thing with delta and others. And it's if you can do that, then you're really getting toward the future of this stuff, which is to use the ongoing pandemic so far and predict the growth rate of future pandemics. And that's, I think, pretty exciting and pretty interesting. We'll see what exactly happens. I don't know, I don't know how it'll hold up in the future because, of course, new mutations will occur on a new genetic background, which is kind of the point I want to bring you to here, which is to say that the bigger problem than all of this is that almost certainly the genetic background a mutation arises on is tremendously important. And we call this effect epistasis, but basically the idea is certain mutations in combination may have a very different effect than they have independently from each other. An example that I think hints toward this is this stuff going on right now with, if you look at B.1.351, B.1017, P1, they all had N501Y, some of them involved 484K and a few others. The California lineage, which is not actually shown on here for some reason, is down here somewhere, definitely evolved L452R, which is also in Delta. They also evolved a number of other shared mutations, and it makes me, it doesn't prove anything, but it starts to hint that there are sort of separate trajectories that the virus can go on depending on which mutations it gets in the first place, right? So if there's a strong effect where once mutation A arises, B is tremendously favorable, we should see that path occur many times, A to B. Whereas on the other hand, if when A arises, B is no longer favorable because they're sort of excluding each other, then we should very rarely see the alternative. And this is, I think, going to be a really important thing to get at. It's something actually I think Vladimir is working on right now, or you guys are working on collectively. So I'm very excited to see what you come up with. I think it'll be the future of a lot of these stuff because, like I said, there's pretty good evidence that there is epistasis. Certainly the protein structures will be affected by multiple amino acid substitutions. So we think this should be true. And the cool thing about epistasis, I know it sounds like it just makes the problem harder, but to sort of plug it and tell you why I think it's cool, is that it makes us more predictable, right? So if we actually understand those components and you see a certain mutation arise, we can do a much better job of predicting the next mutation if we know how those two interact, right? And to the extent that evolution becomes predictable, this is tremendously important for forecasting and understanding the virus in long term. Okay, with that, I need to take another small break. I'll be right back. I need to switch my network because this is kind of fading in and out. I don't know if I'm coming through clearly or not, but I'll log back in just a second, and sorry about that. So give me 4 minutes while I switch this network. Yes. All right.
Okay.
Hello all, sorry about that. How bad was that, by the way? The Zoom keeps telling me that my connection is unstable. I have no idea if...
That was totally fine.
It's fine. It's
Oh.
more than perfect.
Oh, okay. Then I don't know what to tell you. I'm on a different network now, so I guess it won't matter, but that's okay. So nice to see you all again. Thanks for having me back. Okay. With the second part of this, so first of all, if you have questions, I'd love to take them now. I know that it was kind of a whirlwind tour of what we think the right way is to start looking at selection in this in SARS-CoV-2 scale phylogenies is. I don't think anyone really knows, right? There's been a number of approaches that people have tried, and I think the sort of the more typical long-standing versions of this stuff take too long to compute, so they're not really useful in my opinion, and at least not if the goal is to do something that's sort of actionable now in the context of public health, right? If we want to study the pandemic years later, then that's great, but it isn't. I don't think that should be the goal. I think the goal should be to figure out how to use genomic epidemiology to make real intelligent decisions right now. So if you have any questions about that, I'd love to take it. Otherwise, I can move on to the second section here.
Well, yeah, I guess I have one.
Sure, yeah.
As usual, yeah. I'm just curious how robust these methods to the frequency of the mutation. I mean, it sounds kind of intuitive that the more mutations you have, the worse situation gets, or is it?
So,
sorry. I must have misformulated my question.
Okay.
Hey, let me just. Okay, yeah. So for some mutations, they are getting reintroduced again and again and again and again, and you do not have like one big good tree which corresponds to a mutation, but you actually have a collection of smaller ones. Yeah, for the mutation of high frequency, or maybe there's some backward mutation on the same site. And I am curious. Do these effects, the fact that there are like many similar mutations in different parts of a tree instead of one big chunk of a tree being corresponding to a mutation, does it influence like any of the methods at all?
Yeah. So first of all, that's a really good question. And it's probably a lot deeper than I'll be able to go into right now in great detail, but yes, this must matter. Right. So, for example, the mutation at D614G is now fixed. Right. So I don't have the ability to go look at selection at that site because it happened basically the one time and went to fixation. So if I use this sort of like subtree comparison approach, it doesn't make any sense. Right. That can't possibly work. I think that's what you're asking. Right. Is that what you're asking, more or less, Dimitri?
Yeah. So. Yeah, and for the mutations where we have like some differences, let's say one was introduced purely in France and all the France is like in this mutation, and the other sporadically appears at some places and maybe disappears, or maybe something. Other results which analysis show differ in their exactness. Not sure if it is even a word or it basically doesn't matter.
Yeah, if it's just too noisy with these sort of like epidemiological chance events that occur. So far, for Rojo and the Sega stuff, it doesn't look like this happens that often, but they've mostly worked at the earliest parts of the pandemic up to the very emergence of Delta. In that time, only about 30 mutations got to frequencies above like 5% of all sampled viruses. So that on that scale, it must be a fairly weak effect for the vast majority of mutations. But I think a more promising future that is something that I'd like to talk to you guys about as we move on with this is, at least on a fixed tree, it's actually pretty easy to jointly estimate the mutation rate and the effect on growth for a given site, right? So if I fix the phylogeny, obviously if there's variation in the phylogeny itself, or there's uncertainty in the phylogeny, I do many times, it can be expensive, but things like that are very possible to do, and I think would probably be a little bit more robust than the approaches I've seen used so far, and I don't think they're that hard to do, quite honestly, so I would like to do these things. So we're working on these things, and I would love to talk to you guys more about it. But I think you've brought up a good point, which is that the kind of simple things people have done so far probably have lots of assumptions that will be violated or already are violated in some useful way. Yeah, cool. All right, cool. Let me tell you guys a little bit about our work on recombination, and then I'll speculate at the end again about just what I think the future of this this kind of analysis will be, and I'd love to get thoughts and feedback about that. I think it will be the most interesting and exciting time in this sort of stuff because it's just such a different kind of data and a different way of thinking about the data than I think has been typical of academic research so far. So I like to subtitle this, right? So recombination in SARS-CoV-2 is, it's not super common yet, but it's certainly happening. And the subtitle I like to use for this is when the trees aren't actually trees, right? So we spend a lot of time trying to infer a phylogenetic tree. Recombination is not a tree. Like when you have recombination and you detect it, it means you have a network. And it's a little bit of a different problem. I think the way we've been doing this probably works pretty well regardless, but I think the future of this will have to deal with this stuff a little bit more aggressively. And so to show you a little bit about how recombination works, you may remember way back at the beginning today and many times before, I said, well, there's this thing that goes on, which is that there's more subgenomic RNAs at the back of the genome. Keep that in mind, it's important. The way it works in this case is you get a human, and this is a human cell in theory, and there's two genetically different forms of the virus, there's red and blue. And red and blue, are both replicating in the same cell at the same time. And then red, the polymerase in red jumps off, it falls off, and it jumps onto a different template. And it goes to work back on that template and gets to the end. And at the end of this thing, you get a mosaic virus genome out that's half red and half blue. So this can happen. It does happen. It's demonstrably true that this is happening. But some important questions are, well, how often does this happen? If it's happening very frequently, is there strong selection on these things or not? Okay, so if you zoom way out, and instead of looking at SARS-CoV-2, we just say, let's look at all beta coronaviruses, or most beta coronaviruses, and then we make a kind of tree out of different regions of the genome. So this is the first third of the genome makes this tree, the second third of the genome makes this tree, and then the last third of the genome makes this tree. And what you can see is that these aren't the same, right? The topology is actually changing across this landscape, which strongly suggests that some amount of recombination is occurring, right? So we see up here, this red strain Wuhan 1 is grouped with these two, but down here, those have moved kind of toward the center of the tree, and Wuhan 1 is out on its own up here, which would suggest that there's a recombination edge somewhere right there, more or less, between these two nodes. So cool, okay. This is certainly a thing we expect to be happening. It's really easy to see on these longer time scales because presumably the recombination events are occurring between genetically extremely distinct virus genomes, so it's quite easy to see and understand. For SARS-CoV-2, it's a little bit harder, but I think it's really important because in SARS-CoV-2, this could drive the evolution of new variants of concern. So sort of a recombination event where there's a fitter front half of the genome and a fitter back half of the genome that brings those two together could produce something really important for the future of the pandemic. For the reasons I talked about a little bit at the beginning here, this can distort phylogenomic and phylodynamic inferences, right? Because if your tree isn't really a tree, but you sort of force it to be one, it can mess with your inference downstream. So it's actually important to have a handling on what this looks like. So there's been a couple different ways I've seen of doing this sort of a little bit more rigorously in the ancestors of SARS-CoV-2. So again, here they're looking at beta coronaviruses. This is a paper from, I think his name is Bobe, and Howard Ochman and someone else who went through this. Again, I can't tell you because my captions are cut off, but who did this sort of thing where they're looking at the ratio of apparently homoplastic mutations versus non-homoplastic mutations and just sort of bins across the genome. The logic basically being that if there were a lot of recombination, we should see an excess of homoplastic mutations in certain parts of the genome and then dividing by the rate of non-homoplastic mutations is really useful because it can correct for the local mutation rate in theory, right? So If you do this analysis, what you find is that the spike protein region has an enormous excess of apparently homoplastic mutations across all the virus genomes. So suggests that there may be an excess of recombination here. I think one thing that the paper didn't really deal with is the possibility that selection is actually favoring those homoplastic mutations at some rate, and they're actually occurring independently and then strongly selected such that you see them in many cases. But that's neither here nor there. The overall signal seems consistent, which is the bulk of these homoplastic sites are in the back part of the genome. There's a much more sophisticated thing from Felix Muller and I want to say Trevor Bedford's group that was looking at sort of Bayesian networks. And I will not go into the math of all this because I'm not trying to understand some of the math for all this, but the basic idea is that if we take this problem and we cast it as a sort of ensemble of trees problem, where I'm trying to compute the likelihood of a site under an ensemble of trees, I can start to pull out where these recombination events have occurred and sort of look at them in time, going backwards in time on my tree. And so they did this. They did this for a number of different genomes, and they also did this for several different endemic human coronaviruses. So there's 4 or 5 different endemic human coronaviruses, 4, I believe. And they did the same sort of thing. What they're basically seeing is, as of before, you see this excess of apparent recombination in and around the spike protein region. If we show that a little bit differently here, we're showing it for 3 of the endemic human coronaviruses. What you can see, these cause the cold, by the way. These are sort of endemic. You've been infected by each of these probably at least once in your life. And if you look at the relative recombination rate, so this is expressed as a ratio, where we're looking at sort of spike versus everything else would be green, you see that the rate is something like 3 or 4 times higher than the rest of the genome that they get, right? And so they're seeing this very consistently in each of these beta coronaviruses, with the exception possibly of, this is subunit 1 over subunit 2, so it's parts of the spike protein. That doesn't have the same consistent effect, but the spike definitely shifts up as far as we can tell. Okay, so that's great, right? We have all these really cool sort of deep time approaches. When you have lots and lots of mutations on a relatively sparsely sampled tree, then we have good ways to think about finding recombination, or at least reasonable ways of finding recombination. But the problem with all that is that these really aren't going to be the way you want to do it if you're going to work with stuff within coronavirus. And the reason for that is that you just don't have a lot of mutations to work with. And so to be able to find these things well, you need a better approach. And not only that, but it should be like these deep time things, it only requires 1 or 2 recombination events to be able to pull it out, because that one event has enough genetic differences that it's very obvious, right? For SARS-CoV-2, which emerged in human populations basically the one time and has acquired all of its genetic diversity since then, this is a much harder problem. And so the problem basically comes down to, well, you need to be able to see recombination. So whatever we do is going to be highly conservative. In fact, the vast majority of recombination events probably occur between genetically identical viruses, and we can never see them at all because it doesn't induce any difference in the haplotype structure. Yes, it is technically a network, but it is indistinguishable from a tree, so who cares? And so we don't worry about it. But what probably happens with in some cases is something like this, where you have a split in your tree, and there's these 2 strains of the virus, and they are both co-circulating in some large human population and acquiring mutations. So this acquires 2 red mutations. This one acquires 2 blue mutations. After that amount of time has passed such that they're able to acquire these mutations, they need to transmit then in the same human population. I'm showing this in California because that's where I'm at. So in other words, they have to be distinct long enough to acquire mutations and stay transmissible and extant in human populations. Then they have to transmit within the same population. Then they have to co-infect the same individual and recombine there, right? So in other words, they have to not only co-infect the same individual, they have to actually be replicating within the exact same cell in that individual. And then that individual has to transmit that mosaic recombinant virus back out into the world. And eventually it has to get transmitted all the way to a sampling and sequencing center such that we're able to actually see this. And so not only do we have to sample that, we have to actually sample the ancestors as well. And so this is kind of a lot, right? This is a tall order. And so what this means is that no matter how we look at this problem, we are almost certainly going to have a very conservative estimate of the actual rates of recombination in human populations, right? Because there's just so many steps, right? Like right now within human populations, there's basically one strain of the virus in most places. So even this first part is unlikely to happen. Assuming that does happen, and it certainly has at certain times, then they have to get to the same individual. Most, if the infection rate in your community is high, co-infection is much more likely because there's many people with transmitting the virus. But that's not always true either. Sometimes you have very, very few infected individuals, in which case co-infection should be incredibly uncommon. And then finally, you have to actually sample and sequence a thing, which in the United States, in the UK, and a lot of Europe, it's pretty likely. But in many parts of the world, you would basically never actually sample that virus because we don't have very good sampling. So these are our problems. And so that's hard. The other main challenge is that the data is enormous, right? The same problem we had for phylogenetics applies here as well, which is there's just too much data for the conventional approaches. So, the standard thing that people would typically do in the past would be to use like the program RDP, which looked at every possible trio of virus genomes. So it's a choose three problem. And it would say, okay, well, are there cases where one of the three shares chunks of the genome with one of the other? Which sounds good until you realize that that's a very expensive operation when you have millions of genomes, right? If you're at 4 million genomes, the choose three problem is, I don't know, probably 10 to the 20th or something. Like it's enormous. Like there's no way that you're going to do it. So we needed other approaches. And what I've seen people do so far in general is try to reduce the search space, right? So what they do is they consider specific nodes on the tree. And what I mean by that is, in this case, there's been a few versions of this. I'm just showing one of them. From Vandesberg et al., it was Katja Kohl's group. And what they basically did, and hey, my citations are on here, that's great. So what they did here is they subdivided the tree into little clades, right? So 20B2 had a set of defining mutations, 20B1 has its set of defining mutations, 20A1, et cetera. And then if we contrast them, we say, okay, well, let's look at 20B2 and 19A1, well, then there's, I don't know, maybe 10 mutations along these branches that distinguish these. And so if we see a virus genome that has some combination of the two, it would suggest to us that it may be a recombinant, which seems pretty reasonable. And this is their little table they came up with of all these sort of clade defining mutations. And so the nice thing about that is that this data matrix is like 25 columns long. And so it's just not very hard to take every single virus genome and say, do you have a set of mutations consistent with one or the other, and just very quickly go through all of them. So that's great. The problem here, of course, is that this tree is not really quite the right representation of the data. And actually, there are many little branches that come out on these. And so they're obviously going to be a mixture of mutations among different viral genomes. And so that's one way this can go wrong. The other way this goes wrong, and I think is probably most important, is that we've already shown you mutations are highly recurrent in these genomes. And in fact, this position shown here is 11083 is almost certainly the most recurrent position in the entire genome. And so I get really nervous when people use mutations like that, assume they can only occur once, and then say it's evidence for a combination. I think it's very unlikely this is an appropriate analysis for these data. So an alternative that I've seen quite a bit of is using very few samples. So there's people that have used RDRP, and this is a parsimony-based ARG called QARG or something like this, where basically what they're looking for is a case where if we subset our data down to say 50-ish genomes, we can analyze it pretty deeply. And we're looking for a case where we see some inconsistency, some network-like structure in an otherwise tree-like thing. And so this might work in some cases. I think it has some things that are tricky to deal with, but the biggest issue with this is that, like I said, recombination should be rare. I don't think you're going to find most recombinants this way because you've subsampled your data so much. Most recombinants are probably very rare. Obviously, I would expect that we don't see a lot of the potential recombinant genomes if we do it this way. And so finally, another version of this reducing the search space, which I think generated the most convincing examples of recombination, was to condition on a specific very long branch in the tree and look for mixtures of mutations on that. In this case, they used that branch, that alpha lineage, B.1.1.7. And if you do that, and you throw all your mutations along your tree, up here and down here. And then in the middle are the potential recombinants, these four. What you can see is that along the top here, this particular individual does not match these, right? So they have different mutations at almost every position up until somewhere in here. And then it switches, right? So right somewhere between 16176 and 20410, we see this, or sorry, that's, yes, we see this switch over here actually. Sorry, talking to myself. The position that you actually want to look at is over here. So it's somewhere between 21255 and 23063. There's a switch, and then suddenly these four in the middle switch over to matching this top one. So this is an example of what we think recombination ought to look like, where one of your these four in the middle are related to each other. Their haplotype matches the top for half the genome, the bottom for half the genome. It would strongly suggest there's a recombination event somewhere in there, which is great. But again, it reduced the search space. You need a very specific recombination event for this to be useful. And so we were staring at this problem, and I said, well, we wrote this program Usher, which is, outrageously efficient for what it needs to do. What if we made the problem a lot harder? So rather than an n problem where there are n possible nodes in the tree to search, let's make it a type of n squared problem where we're going to search a ton more nodes. And the way we basically think about doing this is we say, well, let's look across the tree for a signal of recombination. So what recombination ought to do is produce an unusually long branch in my tree. And so if we do that and we search across the tree for things like this, then we might look at this branch here, F. And F, to clade F here down to this branch, to this node D, requires 4 mutations. So that's a pretty long branch that's potentially indicative of a recombination. A hint I'll give you in advance is these are all near each other in the genome as well. They're at positions 10, 11, 13, and 14. So that's something recombination ought to do as well. If we do this, then you can, the solution we came up with is a totally brute force solution, which is find these branches and then exhaustively subdivide the genome. So find every possible break in the genome and replace those 2 halves of the genome, or we also allow 2 breakpoint models, but this is only showing 1. Replace those halves of the genome and see if we can find a better configuration, a more parsimony optimal configuration. So this particular case, we say, oh, well, we found a case where this only requires 1 mutation, so that's a good sign. But actually, this is the right answer here in the middle, where we find a case where if we subdivide between positions 9 and 10, then the parsimony score is 0, and it's very consistent with what we're looking at. So what we're finding is potentially an edge in the genome where a recombination event occurs and some portion of the genetic material from one is transmitted to the other, or vice versa. Okay. And so that's the idea, right? So let's just brute force this, make it a computer science problem, and we're going to do it all in parsimony. So in parsimony, the other thing is we need some way to determine what we think is a significant improvement. And the way we did that was to simulate mutations that we think are consistent with the mutational process in SARS-CoV-2, and just throw them back down on the tree and start asking how commonly we could see parsimony score improvements using our approach when it was a mutational model. And you can use that. You can say, well, a parsimony score improvement of 3 to 2 is actually very common. You're very likely to find that. But a parsimony score improvement of 3 to 0 is actually very uncommon. And so you can use this to get a very an approximation to the probability of observing these. Okay. So what do we do? Well, we wrote this and we ran it. And what we discovered, among other things, is that so the first thing you do is you ask how sensitive it is. So we simulate what looks like real recombination events. And we do quite good, right? So we recovered about 93% of the total recombinants that we simulated. And what I'm showing you along this axis here is the minimum starting parsimony score, which is another way of saying how genetically distant the 2 parents of the recombinant node are, right? And so if you're over here and we're at 3, 4, 5, 6, 7, that's a relatively low parsimony score. And so you don't see the same sort of effect where, what that means is your parents are pretty closely related to each other. And we actually do a little worse. So in red, I'm showing you the subset that we simulated that we don't find as recombinants using our approach. And in black are the subset we do find. And so we actually do very well when the parents are very genetically distant. We do a little less well when they're closely related, as you would expect. The same is true over here, where we have this minimum starting parsimony score, and but in 2 breakpoint models. And so with 2 breakpoints, we actually typically do a little worse. It's a harder problem. But again, it's overall pretty darn good. The second thing that I'll show you is if we, on the top, I show you what we simulated, and you see that it's pretty much a flat, as you would expect. We simulated from a uniform distribution, so it's pretty flat. On the bottom, I'm showing you how well we did. And in each of these cases, we don't really detect the stuff at the very far edges of the genome, right? All these are simulated, we don't ever find those. And so clearly the edges of the genome are going to be hard for us. And probably the reason for that is it's, if you put a breakpoint here, it's very unlikely that there'll be a mutation further to the right that distinguishes the two parental haplotypes the way you'd like it to. Okay, so we think ripples is going to work well, but we're still working on it. And so we use ripples, and we searched a 1.6 million sample tree for recombination. This took about 3 days in total to run the whole thing on a pretty beefy server. And then we did some extensive QC, and I'll explain why in a second. We found 606 unique recombination events. That is, so those are nodes on the tree where we think the descendants are recombinants. Approximately 2.7% of the samples are detectable recombinants. So if we take those 606 nodes and we count up all of their descendants, divide by 1.6 million, it's 2.7% of that 1.6 million. And if we estimate this parameter, R over M, which is the amount of variation in a given sample that occurs as a consequence of recombination, divided by the amount that we think occurs as a consequence of mutation, you get a very, very small number. It's much less than 1% of sites. So while recombination is certainly happening and it's quite detectable, the effect it's having on the tree is relatively small so far. And so I think a basic tree structure is probably okay, but we'll see. One thing I do like to point out, so I said we did extensive QC, it's very necessary. So an unfortunate feature of the way these data are produced in this highly disaggregated way is that lots of weird stuff happens. So for example, you get something like this where in a simple multiple sequence alignment, we might call that as many as 5 or 6 mutations, right? But, or more, I don't know, I should count them actually, there's 8 there, I guess. But what's probably just happening is it's an assembly error that's occurred a few times because you have an extra A on this side and one fewer A on this side. And so we don't think this is quite right. The same sort of things happen kind of toward the edges of genomes, which tend to be hard to assemble. So you see these kind of falling off into gaps. Presumably it's a large indel or a gap, which has been assembled incorrectly. And so we see these kind of like mutations that appear to support recombination, but certainly don't, as far as I can tell. Okay, but what are some predictions we would have? So one prediction I would have, if I was finding real recombination, is that there ought to be an excess of geographic overlap, right? So if I throw down my recombinants at random on the tree, what I would expect is that what I would expect to see is that there's some overlap, of course, because you can randomly select samples in the same region. And so if we do that for the background rate, and we ask how much overlap there is, so this is just the set similarity, where 0 would be there's no overlap, and 1 would be they're exactly the same, all of their descendants are in the exact same region. In gray is the background rate. So most of the time you actually don't get any overlap or not much overlap, but it varies quite a bit. In blue is what we see, which is a ton more overlap, right? It's highly skewed to the right. And so we feel pretty good about this. It strongly suggests that a lot of the signal we're seeing is consistent with real recombination events because the two virus genomes that we're seeing must have co-circulated in the same place at the same time. If we go on to the other side here, the same sort of thing works if you look at the overlap in time, right? For two virus genomes to be recombinants, or for a virus to be a recombinant of 2 other strains, those 2 ancestral strains must exist in the same place, but also at the same time. This is trickier because the vast majority of sequencing was done at the time we did the analysis in the 6 months prior. So there was often quite a bit of overlap by chance, but we still see a pretty strong excess of overlap in the recombinants we think we detected relative to the background set of simulated recombination breakpoints. So the point of this is to say that these are consistent with our expectations. That there's an excess of overlap in time and space relative to a random sampling strongly suggests that we're getting something reasonable out of our data. Okay, some example detected recombinants. So I'm showing you the same basic figure as before, but just what we got out of it, where the middle is the recombinant node and the top is the acceptor and the bottom is the donor. Those are exchangeable, but don't worry about that. The point is, if you see this very nice pattern where the haplotypes are literally identical for quite a ways. There is some recombination breakpoint between these two SNPs. And this is a good example of a case where they're actually really far apart from each other, right? This is 11704 to 20080 or whatever. And so it's a really big window in here. And then they're identical across the top again. The same is true down here. It's a nice long one. And then a 2 breakpoint case here where it matches the acceptor for the first third of the genome. The middle half of the genome matches the donor, and then at the last little bit of the genome matches the acceptor again. So presumably this is a 2 breakpoint model. Okay, so that's great. All right, what about the spatial distribution of recombination events? I think this is maybe the most interesting is, if we look at those really deep time things, it was obvious there was an excess of recombination in and around the spike protein region. But that might also be a result of natural selection, because if the spike is also the main site of adaptation, presumably shuffling haplotypes in that region could be adaptive. And so it may be a bias. Whereas when we look at recombinants in a very, very, very small chunk of the tree, that shouldn't be the case. And so what we find here when we do this is that, and I'm not plotting, a gray bar is the confidence interval or prediction interval, where we believe a recombination event has occurred somewhere between those two edges when we do this. And so the first thing I want you to point out here is that those are not very accurate, right? They're big chunks of the genome. It's hard to know exactly where recombination breakpoints occur. But even so, if we just show you, this is just the density kernel of the midpoints of those regions, you see this effect where it's pretty darn clear that there's a lot more of these in the back third of the genome where the spike protein is than in the rest of the genome. This line is just from breakpoint analysis to show that, yeah, it does subdivide nicely into 2 different regions. The back part of the genome has an inferred recombination rate about 3 times higher than the front third of the genome, front 2/3 of the genome. So we do think, just by neutral processes, that there is a ton more recombination in this part of the genome. And this, again, is consistent with this idea that I mentioned earlier that I don't have a figure from now, that this could be because of template switching onto subgenomic RNAs. Remember, the way the virus genome works is there's these long subgenomic RNAs that are used to produce the proteins. Well, if you have a template switch event, presumably most of the time you just end up back on the exact same template you were on already. But if there's a ton more partially homologous molecules from this part of the genome, then it should be that there's an excess of template switches onto these as well. And that we think this is consistent. This is what my virology friends tell me I'm seeing. So I think it's potentially really important, because it suggests that not only is recombination occurring, of course, but the more fundamental thing is that recombination will disproportionately produce new haplotypes and variation in and around the spike protein, which is something that we think is really important to think about. Okay. This has likely impacted the pangolin naming system. So just to show you an example of this, this is a case that we see here where where we see this particular lineage, B.1.355. It appears to be a recombinant from a node within B.1.371 and B.1.595, where this lineage inherits this set of mutations and this one, but none of these that occur in the back part of the genome, because we see this one instead. And so we think this and many others are probably recombinant lineages that are arising in SARS-CoV-2. Okay, maybe the most important and the coolest thing about this is that Ripples has real-time capabilities. So we designed this to work on the exact same data structure that we use in Usher. And the reason that matters is if you have a new sample that you want to know is potentially recombinant, you don't have to reinfer an entire tree and do the whole analysis from scratch. You can actually just add that sample to our existing tree. Right now, that takes about 20 seconds for a 4 million sample tree, but we'll see how fast that can be in the end. And then you can just do this the Ripples analysis, which is to search the entire tree for potential recombinants just associated with that sample and its ancestors. So this just takes about 4 minutes total, something like that, which is remarkable, I think, because it means in about 5 minutes I can tell you if your sample is probably a recombinant sequence or it has recombinant ancestry. So it's pushing toward what we think is really important, which is getting really timely results back to the people that need to understand those results and to figure out how to tell them this stuff. We're going to skip that because I don't think we'll have time today. It's 9:50 already. So I just want to leave you with something I mentioned on the first day that hopefully you'll appreciate a little bit more why I think this is so important, and maybe we can have a short discussion about this, which is, what do I think the future of this stuff is, right? So what's going to happen is that this is going to transition from a very academic exercise, which is what it's been, into really full-blown public health and medical thing, right? People are going to use pathogen genome sequences to make medical decisions and public health decisions, and we need to get better at that. And so what I think that looks like is that a sick patient comes to a clinic, the virus genome gets sequenced, right? So like we won't even think about it. Like a person comes in, we don't even necessarily know why they're sick, although we may know from testing. We will simply sequence them no matter what. And then once we figure out what it is, and there's lots of metagenomic approaches for that, like ID-seq and others, a tree like the one I have already for SARS-CoV-2 will be ready for that pathogen, and we'll add the sample to that global tree. And this, again, this will happen in seconds, right? The feedback needs to be almost that immediate. And then we'll instantly display that for public health and for doctors. And so pushing toward this idea that I've been trying to summarize in a simpler and simpler way, which is everything needs to be faster. So, everything we've been doing to this point has been very academic, very post hoc, where I say, oh, okay, well, let's just spend months analyzing some small data set that we have. That's not what we're doing anymore. What we need to do now is be able to use millions of sequences as they're coming off the sequencers in real time, which is really going to be a lot more of a computer science and an engineering problem in some ways, but it's also, I think, really important for getting the stuff to the final form that it's going to take. And to give you a kind of an applied example of what that means, why do I care about this strain-specific annotation? Well, I care about this because we may already know things about the relatives of that new sample from this patient. For example, if I know that sample D here is a known antibiotic-resistant bacterium, then I can give you a pretty good guess that sample E might also be an antibiotic-resistant bacterium. And that tells me something about what treatments would make sense for this individual that has this. The other thing that needs to happen is we need to get medical records linked up to this, right? So it's not enough to have the annotations we already know about. What we need to be able to do is to generate new evolutionary annotations. And so the way to do that is to say, okay, well, once we have this genome sequence from E, maybe I don't already know that there is antibiotic resistance genes. When I look at the treatment my doctor gave them, which is some antibiotic, if they're non-responsive to that antibiotic, that annotation needs to get back into my tree. I need to know that the relatives of E may not be responsive to this particular antibiotic, right? So we need to be able to do that also in real time as it's coming online. Okay, and I think that's about all the time I've gotten about what I've got for you. So I'd love to take some questions here for the last 5 minutes and we'll get out of here, but thank you so much.
Thank you, Russell. And I guess I will leave a role of advocate of a devil to someone else, because I think I've asked too many questions.
Oh, you're doing great. Someone's got to ask the questions, man.
Yeah, I'm telling you the same stuff. Come on, fellas. It's cool.
Nope. All right. You sure? All right, Dimitri, go for it. You got questions.
Well, actually, the most striking feature is for the last hour. I do not have real questions because, yes, I agree. Well, I might just consider that maybe, just maybe, there is some trade-off between, exactness and computational speed. I mean, what do you think? Where can this come from?
Yeah, so more complex models of nucleotide substitutions might do better. It's going to do better in cases where, if you just use something like parsimony and you have a tie, so there's two different places a sample could go on my tree, and it requires exactly one mutation. If I had a more complex substitution model, I might know that one of these sites is extremely highly mutable and the other one is not. And in that case, I would say, okay, well, it's much more likely that the more highly mutable site has occurred again than that this thing has independently evolved this not particularly mutable mutation, right? So examples like that are going to be really important. And it's something we've been thinking about and working on more and more because we know it's going to be, we know this is going to be a part of the problem that we want to solve going forward, which is how can we do something like maximum likelihood that doesn't take insane amounts of time. Imagine that we were doing something where we didn't care about all the positions in the genome that almost certainly didn't mutate, right? Our branches are so short that I'm not going to do Felsenstein's pruning algorithm across every single site. I'm just going to pick the subset that probably did change and then use that, right? There's other ways to do it, but that's one of the ways.
Yeah, sure. Just if we are to generalize it, let us just have some priors and which are interesting for us and which are, well, influential for us, and just focus on that, them, and priors being like chosen sites or maybe something else. I'm not sure, but yeah, I'm getting your point. Yeah.
I think that's one of the ways this will become a more general problem, but there will be others as well. I don't think that the truth about it is, and I think I showed you this, guys, about a week ago, was right now, parsimony is doing better than all of the best-in-class likelihood stuff. And the reason for that is probably not that it's a better model. The reason for that is that I can search a lot more of the tree than they can, right? I can just try more tree moves than they're able to do. And so at the end of this whole thing, I will have more chances to find the right solutions. What we'd like to be able to do ideally in the future is do something as computationally efficient or nearly as computationally efficient as what we're doing. But as you said, add some priors, generalize the model, think of the ways that we can get the little things that we were getting wrong right going forward. And we're working on it.
Yeah. Okay. Well, thank you very much for the talk, like usual.
Sure, no worries. No one else? That was too easy? All right.
Yeah. Thank you very much, Russ. It was really exciting, at least for me, and I think for Dmitry.
Excellent.
Yes, and thanks everyone for attending these lectures. And we are done for this year, at least.
Hey, all right, congratulations.
Hi, everyone. We're still here.